This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 55 for October 23rd, 2009. Hey everybody, it's Vincent Racaniello, and we're broadcasting from the TWiV studio today in New York City. And right here with me at TWiV is Dick Despommier laughing his head off.
Hey, Vince.
What's so funny?
I'm just amused by all the people that showed up.
And also Jason Rodriguez is back.
Hi, Vince.
How you doing, Jason? Fresh from the bench.
XMRV's making me a popular guy.
Isn't that great? Doesn't always happen, you know.
It's true. It's true.
Oh, run with it, kid.
Run with it, baby. And from remote locations around the globe, Western Massachusetts, Alan Dove.
Hello.
I
Good to
think
be back.
He's there. Very good. And down south from Florida, Rich Condit is back.
Howdy. Great to be here.
Howdy. Howdy. Hey, I saw a picture you put up on Facebook. He was on the golf course and there was an alligator crawling. What was that? One of the holes, right? The seventh hole or something?
Yeah, it was seventh hole, seventh fairway. Yeah.
Seventh fairway.
Yeah, he was on his way from Lake Alice, which is on campus, to one of the water hazards on the golf course.
How wonderful.
That's great. We don't see that up here, so I thought that was pretty
I presume
cool.
he feeds on golf balls.
Wait, I have to ask something. Am I the only person in the world now who's not on Facebook?
Yes.
I think Dick is not.
Yeah, I am.
You are?
I am.
Oh, God.
Do you actually use it?
Of course not. What do you do with it?
All right,
I
so
already
if you
know
have
what
to
my face
ask,
looks like.
if you have to ask, how does the saying go? If you have to ask,
You shouldn't have to.
You should join, I guess.
something like that. If you have to ask how much, you
That's right.
can't afford it, and
That's
so if you have to ask, you shouldn't join.
Well, I think everybody's on Facebook now because my mother now is a Facebook fan, and she always makes these comments whenever I say, "Oh, I have to go into lab and work on XMRV." She says, "Be careful."
She's wise. Mothers are always wise, you know.
You bet they
That's
are.
true.
But you know what,
But there's
Jason?
a lot of people on Facebook.
You might be seropositive already,
I hope not.
but you could check, right?
I can check, but I really hope.
Well, XMRV is going to be one of our topics today, so why don't we get right into it? Last week or 2 weeks ago, a paper came out in Science. So if everyone remembers, Jason was here a few weeks ago. We talked about XMRV, this new retrovirus, and prostate cancer. And I think the week after you were on the podcast, you came in my office, you said, we got to do another one because
There's
something
going to
just
be a
came
paper
out.
coming out. Yeah.
We went to a meeting and heard about this.
Yeah, as a matter of fact, I have the paper with me right in front of me. That
Oh, wow.
was the confidential under embargo. So before this chronic fatigue paper came out, basically CDC says, or the journal says, don't tell anybody about this because it could cause a panic. But now it's out, so
It's out.
we can talk about it plenty.
Embargo. So Alan's very familiar with
That's
the
about
embargo.
when I saw it, too. Yeah.
You get an under embargo too, huh?
I would have talked about it, but I was too tired.
If you,
That's actually one of the two reasons why I don't own any biotechnology stocks. The other one is that, well, I'm not insane.
Right.
That's right. So, Jason, what's going on with this new one?
Well, it's a really interesting paper. From my point of view, it kind of turns it on its head because everybody was thinking, oh, it's associated with prostate cancer, but now XMRV is found to be really strong correlation with chronic fatigue syndrome. And just to show some of the relevance for chronic fatigue syndrome, it's actually a really debilitating disease, and I'm actually reading off the abstract from the paper itself, and it says, chronic fatigue syndrome is a debilitating disease of unknown origin that is estimated to affect 17 million people worldwide. So it's a pretty bad thing. Some of the symptoms include things like being really, really tired, and affects multiple organs and joints, and there's even cognitive effects. And so this study, what they did is that they took samples from CFS patients from an institute in Nevada, I believe, and they tested those samples for nucleotide sequences of the virus, and it came up that about 67% of patients that had CFS were positive for XMRV, while only 3.7% of healthy controls were positive. And it suggests, it suggests that 3.7 of healthy controls suggests that maybe XMRV is actually, it might possibly be endemic in the population. There might be millions of other people that are infected. It just doesn't cause any sort of disease. They might be even carriers.
I don't know if we talked about this when we did the prostate connection, but where did XMRV come from then?
So I guess just to recap a little bit, well, XMRV was discovered in 2006 in a group that studies prostate cancer, and they looked in a specific cohort of prostate cancer patients to see whether there was retroviral sequences, and they discovered this novel retrovirus, XMRV. But they really focused on patients or samples, prostate samples that had this mutation in the RNase-L gene, which we talked about in the last TWiV that I was in, as being an antiviral gene, and most of the samples that had XMRV had this mutation in RNA-L. But since that paper has come out, that theory actually really hasn't held up.
No connection with the RNA
There's
cell
really no
mutation.
So what's the XMRV stand for?
So the XM stands for xenotropic murine leukemia-related virus. Xenotropic, and basically xenotropic means is they gain the ability to jump species. So it's a murine leukemia virus. It looks like a mouse retrovirus, but it's jumped species from mouse to humans. So it's called xenotropic.
So we call that
and it can't go back.
Can't
so
infect
the
mice.
full name, as I understand it, is xenotropic murine leukemia virus-related virus.
Yes.
Which I think makes it the worst named but best abbreviated virus ever.
Exactly.
So another way to state this is that it's a zoonosis.
Exactly.
Yes.
Yep. That's where all new viruses come from, right?
Thank you.
So
most of them, yes.
So this is a mouse virus that jumped into people, right?
Yeah. So you would think that at some point, we got the virus from a mouse.
It's not unreasonable. Hantaviruses do that.
That's
right?
true.
So the idea is that this jumped from a mouse to a human and then spread among humans.
Among humans.
Who knows how long ago it did
But
that?
now it can't go back.
Can't go back. Doesn't want to go back because it's got.
Interesting.
It's interesting. Wow.
So can you trace back to when it might have first appeared in humans?
Well, that's actually an interesting line of thinking, because if you compare all the nucleotide sequences from all the samples that had XMRV, they're very, very, very conserved. So xenotropic murine leukemia viruses in general, if you were to analyze them in mice, they can go back, I'm sure hundreds of, thousands of years, and you can get, huge variation in nucleotide sequences. But with XMRV, it's very, 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 very highly conserved. So it's a recent event, I think.
Got it.
Now, on the prostate data, there was another paper that just came out from this German group that looked at essentially the same thing and found they couldn't find the virus in
no
prostate.
correlation.
Right.
There's still a correlation with RNA-L, correct? And prostate cancer?
In prostate cancer, yes, there's a correlation.
But it just doesn't track with the XMRV in this German study.
Right.
Exactly. And just to give some more background, the RNA-L phenotype that we're talking about, it's actually only found about 13%, I believe, in hereditary prostate cancer patients. Mm-hmm. And of all prostate cancer, hereditary prostate cancer is, just a small subset. You also have sporadic prostate cancer, which is just more or less a random event. The RNA-L mutation itself is just a small part of prostate cancer.
Okay.
Now, from talking with your boss, Steve Goff, so he told me that in mice produce xenotropic viruses related to this.
Right.
Very low levels, 1 to 2 PFU, plaque-forming units per mil. So it's one of those viruses that jumped into people. He said what people often do is they inject mice with human tumor cells, just to study tumor formation, and they always get infected with these xenotropic viruses.
Well, I mean, you also have to think about that, all mammalian cells, human cells, mouse cells, we all have endogenous retroviruses in our genome. As a matter of fact, it makes up about maybe 5% of our DNA, basically. So if you take human tumor cells and you put it in a mouse, that the mouse, any sort of murine leukemia virus can come out, recombine with the human endogenous retrovirus and create a novel virus.
How would this XMRV cause chronic fatigue? We're not convinced yet that it does cause it, right?
That's the thing. There's absolutely nothing in the paper that says that it causes. So chronic
It's just
fatigue.
an association.
It's a very strong association. It's about if you read an article in the Wall Street Journal, which is actually they do a really good job. If you
The
read,
second best science journal in the world.
they actually interviewed the authors of the paper, and they said that about 95% of the patients from chronic fatigue actually seroconverted or generated an antibody response against XMRV. So supposedly almost everyone that has CFS has this virus.
Now, has anybody looked at other conditions, for example, depression, where there's a slight immunosuppression, and you might also, would you see the same thing?
That's a good question. Nobody's really looked.
Or transplant patients for an extreme immunosuppression?
That's another good idea. This study, it's interesting because in the study they actually look at immune cells and whether those immune cells are infected, and indeed they are. B cells, T cells, they all have virus intracellularly, and the virus is coming out of the cells and can infect cells that have never seen XMRV. So they're very infectious. So my take on it is that perhaps patients that have chronic fatigue syndrome, they might be immunocompromised already and might be a better host for XMRV.
Sure.
Right.
Doesn't necessarily mean that XMRV might cause chronic fatigue syndrome. All those studies need to be addressed first.
So over the years, other viruses have been implicated, or the herpes viruses.
Actually, a lot of viruses.
And
B19 parvovirus has been implicated. Enterovirus has also been implicated in CFS. So when you look at the history of CFS patients, you would think that there's something must be not working well with their immune system. So maybe XMRV is really good at taking advantage of that.
Just an opportunist.
So what's happened to all of those previous studies with, say, association of a parvovirus? Are those like out the window now for some reason or another?
I think in the community, yes, it's pretty much out the window. But that's what I've heard. That's what I haven't actually gone and read all these papers because there's actually quite a few. Yes, basically, there's not any strong link to show that or causality for those viruses to CFS.
So
given that history, it seems to me we're most likely with XMRV, we're just looking at another symptom rather than a cause.
Could be. But also, the fact that almost all the patients seroconverted is also very, very strong.
Sure, because they would all have. I mean, there's also a very high rate of other comorbidities with this, like depression. Not to harp on that, but that's something that comes to mind as a potential factor.
Yeah, and then there's also the standard epidemiologic surveys that you do for clustering, and I would wonder about this in particular. Are there clusters of people suffering from chronic fatigue syndrome, or is it randomized throughout the population? I mean, where do you find CMRV?
Well, that's actually was a big critique of the paper. Joe DeRisi from California, who actually was co-discoverer of the virus, made one of his big criticism is that we don't know what the patients were, male, female, what age, where they were, that sort of thing. So
really?
that was another big hole in the study.
So there are 17 million people, and we don't know who they are.
Exactly.
who have the
How do
disease.
they know there's 17 million people then?
No, it's not, this study, they didn't identify where the samples were from. That's what he means.
Right.
So they could have been from one place in the U.S. where there are a lot of mice, right, and they get infected often.
They could have.
So, in fact, I got an email from a guy at CDC who said a major flaw is that no information is provided on either the cases or the controls. So it could be.
Yeah.
You have to do that but that's the next step, right?
But my take on chronic fatigue syndrome over the past, not the present or the future, is that people tended to dismiss it as someone who's just trying to get out of working.
Yes.
Right.
They're just lazy, and they had no.
Yuppie fever, I think it was called.
But I mean, it is obvious. That's like post-traumatic event syndrome, like guys coming back from Iraq, for instance. In the early days of
the Gulf War syndrome.
The Gulf War syndrome was totally dismissed out of hand, and in Vietnam as well. But of course, there's a lot of immunosuppression that goes on during warfare, no question about it. But these wouldn't be those people, I take it.
I think this is a real syndrome, and I never doubted it. I think a lot of people felt otherwise, but having a possible agent just solidifies it in a lot of people's minds, I guess, which is
well,
good.
I think they're
and that's good.
Yeah,
there are a bunch of difficulties in working on it because it's also used as a diagnosis of exclusion.
Yes.
So something's wrong with this person. They're not feeling good, but we can't figure out what it is. We're going to throw them in this bucket.
Right.
So there are undoubtedly diseases we haven't discovered yet that have just been tossed into one of these bins and don't know what to do with them. So this could be a very, very diverse population of actual underlying conditions, and we just haven't dealt with what's going on.
Of course.
I was reading how they diagnose you if you have CFS, and you basically have to wait 6 months worth of suffering and to actually be considered, and you have to have years and years and years of history. So, a lot of these patients that suffer from this disease, they really go through.
But this is not going to be a diagnosis, the XMRV presence, right?
Well, no, not as of yet.
I mean, it may be a risk.
Because you could have prostate cancer.
Exactly.
Or not.
That's right.
I mean, but the thing is, if you talk to some of the authors of the paper or go to the website, they say this is the cause of CFS.
I know.
We were the first to identify XMRV.
Yeah, sure.
Right.
It's too definitive. I don't think the patient should be given false hope. I mean, it's great.
So here's another question.
Well, I think some of the authors are at an independent institute that just treats CFS, right?
Exactly.
Right.
So they've got a very strong stake in the outcome of
and
this.
Alan, aren't they paid to treat the patients?
I would assume so.
Okay. Dick, what were you going to say?
So the other question I would have is, what is the rate of the virus in blood bank sera?
That nobody knows. I'm sure the CDC is working on that.
I'm sure they are too.
And the NIH.
I'll bet it's there.
But I would
Of course.
it's there. If you, even in the paper, in the abstract, they say indicator cell lines following exposure to activated PBMCs or peripheral blood mononucleocyte cells, B cells, T cells, and plasma derived from CFS patients who were infectious. So,
Yeah, they have
Wait,
virus.
so they were only looking at infectious virus in these?
Not all. No, they did PCR too. Right?
They did PCR as well.
Okay.
Yeah. They did
Very
PCR, cell-associated virus infections, cell-free virus infections.
Okay, because one of the obvious possibilities is just reactivating from some compartment.
Right.
That's true.
And then maybe we're all carriers.
Well, I mean, that could be maybe a herpes virus activates this or vice versa. And that's why you might need more than one virus. So
or maybe even immunosuppression activates it.
Immunosuppression.
Sure.
Someone said, what about in the Gulf War, maybe the toxins that they encountered triggered? It could be, because the symptoms are very similar, right?
Right.
Gulf War. So.
Yes.
But I don't think you answered the question of how this virus would cause, or any virus would cause chronic fatigue. Do you have some thoughts on that?
I really don't know, actually. I mean, I can't unless it's the virus is attacking the immune system so strongly and it's infecting B cells and T cells. So if you have such an immune suppression that's causing all these other symptoms. So let's say symptoms such as like arthritis, where all your joints are in pain, that a lot of CFS patients, have. That could be one thing, but.
If you look at a disease that has nothing to do with this, that's called African trypanosomiasis, there are, in the early stages, it's called sleeping sickness. And the reason why you go to sleep is because of the production of tryptophan, which is it's almost a derivative from melatonin.
Is it when you eat turkey you fall asleep?
Yeah, no, it's actually a real phenomenon, but then, of course, when you fall asleep or when you don't move anymore, then your lungs start to fill up with fluid and you get pneumonia. And you don't die from sleeping sickness, you die with it. So maybe this has got something to do with it.
So what I've heard is that if you're chronically infected, you have a constant immune response. And if you know how you feel in the first few days of influenza, where your immune system's cranking out the cytokines,
you get prodromal.
You're doing this all your life pretty much, and that's why you get tired.
Here. Well, there's another thing called cachexin. I don't know if you know that one.
Cachexia.
That's right.
That's where you waste away, right?
That's exactly right.
And that's what
that's
HIV
a cytokine. That's a cytokine also.
Patients display. Cachexia.
Right. Yep.
I want to read this. I think this is. Do you have another question?
Yeah, just one more then. If that's the case, then do chronic fatigue syndrome patients lose weight?
I don't know. Do they?
I asked first.
Some of them, not all of them.
I think that varies.
So they're not cachexic then. They're not.
I don't think they're cachexic. No.
Or anorectic.
No.
No, they don't. Wasting, as far as I know, wasting is not a common symptom.
Nor are they susceptible to other infections as a result of their perhaps heightened immune response.
No, but you know what some of them do? They do develop cancers.
Do they?
And actually, that was a point that I was just about to make to you. What was not mentioned in the CFS study is that out of 101 patients that had CFS and XMRV, also had 20 of those patients had lymphoma. And that's actually very significant because murine leukemia viruses, if you take a murine leukemia virus and infect mice, you're going to get about that
20%.
around that number of mice that develop lymphoma.
I'll be darned.
Leukemia.
They go by inserting next to oncogenes?
There's various different mechanisms, but yes, that's primarily.
But they don't.
Are these lymphoma patients who had been treated?
I don't know.
Treated how, Alan?
Well, chemo.
I see.
Because I'm wondering if maybe, some of these CFS patients were actually maybe looking at undocumented side effects.
That could be a possibility, yeah.
But this virus does not have an oncogene in it, right?
It doesn't have an oncogene in it, and I mean, even a lot of experiments that I've done in the lab, it's just not very oncogenic in general. It's not transforming.
So I guess a good experiment to do with some of these patients is to see whether the virus has integrated into the tumors.
Well, studies like that have been done actually already, where they've looked at sites of integration of where XMRV integrates inside your genome, and not surprisingly, it acts just like a mouse virus. It integrates in random sites of open chromatin and genes that are being transcribed. That's where it integrates, but no specific oncogene per se.
Very neat. Well, clearly,
so how are you feeling, Jason?
I'm feeling okay actually. No fatigue syndrome symptoms.
Right,
So you have been working with this virus on the bench.
Does it work for about 2 years now?
Does it have to be worked under higher containment?
It does. I started working on it in the lab, and I've always, whenever you're working with any sort of virus, you're always very, very careful with it. So we treated it as if it was an amphitrophic murine leukemia virus. So we have our own special way of decontaminating everything that we use. But now that it's garnered a lot more press, I've switched over to the BSL-3 facility in the Black Building. Yes. Voluntarily, yes. Because for one thing, everybody else working in the lab, I want to ease their concerns and their fears.
Sure.
And everything like that.
Well, you're a good guy. Not everybody would do that. Can you define amphitropic?
Amphitropic, so it's similar to xenotropic. So actually, for murine leukemia virus, there's 3 types predominantly. There's an ecotropic, amphitropic, and xenotropic. Ecotropic means that it's a murine leukemia virus that only affects mouse cells. Amphitropic means it's a virus that can infect any cell type. And xenotropic means that it can infect other cell types besides mouse, so it's lost the ability to come back.
So for humans, a xeno or an ampho would be equally nasty, potentially anyway.
Right.
Very neat. Anything else, gentlemen, before we get to this email?
Well, I was just a slight digression, Vince. I was wondering if you ever did that experiment with a rotating student in your lab, maybe take a blood sample before and after they rotate and see if their polio titers go up.
I have not done it, but other labs have, and they do go up.
Okay.
Sure. Most of the viruses you work with, you end up, no matter how careful you are, under BSL-2 conditions, which is just working in a lab with standard protocols, gloves, coats, and so forth, you will get aerosols and you'll infect yourself. So, And so in this case, you've probably infected yourself already, and you have antibodies, I would bet. So you could check.
Vince.
You might want to check. You're looking a little tired.
It would be hard to tell with a graduate student, wouldn't it?
He's not a graduate student.
I'm a postdoc.
He's a postdoc.
Train myself to
He's
stay
going to
up.
be even more tired. All right. I have a letter from a fellow who is a pathogen bioinformaticist. These are such new words, I don't know how to pronounce them. At the Lawrence Livermore National Laboratory. And he says, Dear TWiVers, I was listening to TWiV episode 50 concerning the recent article from Ela Singh's group about the prostate cancer XMRV connection. On the same day, the New York Times reported a study from Judy Mikovits et al. Finding an association between XMRV and CFS. The CFS story has been getting even more attention due in part to the eagerness of patients and advocacy groups to contradict the common stereotype of CFS patients as psychosomatic illness, what some people used to call the yuppie flu. I have no doubt that he calls it CFIDS, is a real physical illness and that it invokes some kind of chronic infectious process. But as a computational biologist and professional skeptic, I suspect that the relationship between XMRV and disease is much more complicated than the people reading these stories are likely to believe.
As someone who's married to a psychiatrist, can I just jump in here and say a psychosomatic disease is a real disease?
Aha.
Yeah.
And
Just in
treatable.
case anybody was wondering.
And
Yeah,
treatable.
no, that's a good point.
That's right.
So even if that turns out to be the case with CFS, that would not make it not a real disease.
well, you still have to treat it, right, Alan?
Oh, yeah, absolutely.
Yeah, absolutely. Although the Singh and Mikovits groups have found strong evidence that prostate tumor cells and peripheral blood cells from CFIDS patients were more likely to harbor XMRV than controls, it's still premature to assert that XMRV causes cancer or CFS. It's equally probable that patients with both diseases have an immune deficiency due to genetic or environmental factors, or an infection with some other agent that allows the virus to proliferate.
Hear, hear.
Even if XMRV can be said to cause one or both of these diseases, the question remains, what is it that allows the virus to establish a foothold in some people but not others? This, of course, is something we understand poorly for almost all viral pathogens, including your beloved poliovirus. How would he know it's beloved to me?
Oh, come on, Vince. That's obvious.
Okay.
You wax poetic every time you talk about it.
I think I did say I loved it at some point. Right?
Yeah, you did.
So I was intrigued when your guest Jason mentioned a mutation in RNase L that he said was present in a large fraction of the prostate tumors that could make the cells more susceptible to viral infection. I went online that night to look up the Singh and Mikovits papers to find out if the RNase L mutation was found in the CFIDS patient cells also. It wasn't, but as it turned out, Ela Singh's group didn't find the RNA cell association either.
I'm going to have to interrupt you for a second, actually,
Okay.
because there is a connection with RNA cell and chronic fatigue syndrome. There is, but it's a very indirect connection. There's actually in some, I forget what percentage, but in some patients that have chronic fatigue syndrome, they have an increased expression of an RNA cell inhibitor that is naturally expressed. So that protein might actually suppress RNA cell itself. So you might actually
The virus can go.
might can go. But again, they look to see whether RNA cell that mutation that was originally discovered had any correlation, and it did not. So that's one of the rationales that they use to
look at this
to
virus,
look at
right?
the virus in CFS patients.
Okay, good. That's good. Thank you. This was a larger, better controlled study than the 2006 paper from Joe DeRisi's lab that did find the association, but I still have to wonder how the two groups came up with such diametrically opposite results. Any comments on this?
Well, I mean, everybody has come to think now that the RNA cell connection is not holding up. Even myself, I've done many experiments in lab now where I've actually, knocked out RNA cell gene and the virus just replicates fine. So, or there is no difference.
Okay.
Yep.
So we don't know. This happens often in science, right? Labs get different results and you never find out why.
Right.
Also, I suspect I wasn't the only listener who was confused by Jason's statement that the mutation was found in the tumors and thought this meant either that the virus was causing the mutation or that the mutation was specific to the tumor cells.
I thought I was clear.
Since most listeners aren't going to run home and read the original papers, perhaps you could clarify. The mutation we're talking about is a variant of the RNA cell gene that some prostate cancer patients are born with and is a possible susceptibility locus, not something that's specific to malignant tumor cells.
Yes, it's true.
correct.
I never mentioned that in the podcast before, but like I said not too long ago, the RNA cell mutation is only found in a small cohort of hereditary prostate cancer patients. So it's a small number.
Okay, so hopefully, Kevin, that clears that up. And then he ends, to end on a more positive note, it's okay, we don't mind criticism, right? I've been listening to TWiV for a few months now and have recommended it to many of my friends and colleagues. It makes my 45-minute commute to Livermore seem to go by in no time at all. I have lots of ideas for podcast topics. For one, it would be great if you did an episode focusing on viral strategies for evading our immune response. I would also love to hear your ideas about iPhone apps for virology research. I feel it's really important to have ways to visualize and manipulate biological data that are both intuitive and beautiful. Thanks for all your hard work on the podcast. So I thought we should read that now because it's connected to the CFS story. And by the way, not a lot of listeners told us to do this story, so I'm not going to mention all of them, but obviously it was important. All right. I think we've done enough with that, right?
Yep,
Anything
sounds
else,
good.
Jason?
Oh, that's about it.
So I'm sure we'll be back with you.
Actually,
with this again.
do
because...
have to run back to the P3 and do some virus experiments.
Okay. Before you do that, you want to give us your pick?
Yeah, it's actually really interesting. I don't know if you scroll down to where I wrote it. it's a really interesting article in the New York Times, and it really has nothing to do with virology, biology, or anything. It's actually a physics article, and it's a strange theory and phenomenon about the Higgs boson. And the Higgs boson is basically this particle that will explain why matter basically has mass, and that's why CERN is being developed to try to find this. And it's now being dubbed the God particle because there's a theory by a few physicists that say, well, the Higgs boson is so powerful that it doesn't even want to be found. Itself doesn't want to be found, so it's actually going back in time to stop the machine from finding itself.
I see.
It's very interesting. You have to read the article.
I could have blamed my experiment on that.
I tried, but no one accepted it.
But it's an interesting article, and
that's cool.
It made me laugh a little.
Very
bit.
good.
So.
All right, well, that's a New York Times article. We'll put a link to that. Jason, thanks so much for joining us.
Great.
And I know you don't have a website, but you can find Jason in the lab.
That's right.
Or in the BSL-3.
BSL-3. Thanks a lot, Jason.
Okay, thanks
Bye-bye.
a lot, Vince.
Right.
See you.
Pleasure. Yep.
Take care.
That's what we got next here. Oh, the next story I thought we should just mention maybe. Did we talk about the ribosome Nobel Prizes?
No.
We didn't.
No.
Good. Well, the 2009 chemistry Nobels were for ribosome structure.
Wow.
Which is an amazing thing. This started a long time ago.
Totally agree.
Right?
Yeah.
And, of course, the winners, now I will mess up some of these names. Venkatraman Ramakrishnan, Tom Steitz, and Ada Yonath. All three of them working independently. They solved the structure of prokaryotic ribosomes. So this started in the 80s, and actually Ada Yonath, who's in Israel, was the one who figured out how to get good crystals. If these are big things, ribosomes are big, right? The prokaryotic ribosomes they solved are a little smaller than the eukaryotic counterparts.
She was a Horowitz lecturer also.
She was, yes. Eukaryotic ribosomes, they have a small subunit that has 30 proteins in an RNA, and the big subunit has 50 proteins and 3 RNAs. Very
That's
tough.
a lot of purification and crystallization to do.
Yeah, and she figured out that if you go to extremophiles, you get better crystals. Halophiles and thermophiles. She's the one that figured this out, and then everybody else kind of jumped in on it. So she's sort of the pioneer, but it took them years and years to get crystals that would give them information, and they finally got it. So now we have these structures, and we can tell, how translation works, how antibiotics work that inhibit bacteria that have ribosomal targets. So anyway, it's beautiful work.
I find this highly amusing at one level only, and that is in the very old days of biology, let's say the 1950s and 60s, as molecular biology was just being uncovered, the DNA molecule was now center stage. All the people specializing in morphology as a taxonomic livelihood, these old school taxonomists that would sit there under a microscope and study the little hairs on the wings of the flies and how many setae they would have inside of each thing, they were all accused of doing poetry at this point because they were just studying the morphology of things.
Yeah,
I think stamp collecting was
stamp collecting. So
Jim Watson's term.
Now we've spent billions of dollars and 40 years of research to learn how to do the morphology of a molecule. And now they're back to basic morphology again. So no matter which way you look at it, excuse me for the pun, you still have to see it to believe it.
Well, Steitz says that he never thought that it was anything more than an insight into the workings of biology. So
form follows function.
So this is a deserved prize, right? Everybody
Absolutely.
agree?
Oh, yeah.
Sure.
Yeah.
No question.
What I like about X-ray crystallography in general, and this in particular, is that there's people have been thinking about this and working on this for, as Dick says, over 40 years, trying to understand what this thing is, and you must over that time build up an image in your mind of what the ribosome looks like.
Yes.
And then finally to see it.
That's right.
Must be really magical.
Tell me about it.
And see whether your image of what it should look like is really what's there. It must
my image was always a flying turkey. Because
you mean a plucked flying turkey?
I have to show Dick. We have a picture in our textbook of this darn thing, and it looks just like a flying turkey.
Actually, I hadn't thought about that. Yeah, you're right. It does look like.
You see it? I don't know if you're looking at a picture of it, but here, let me show you.
No. But I know what you're talking about.
See, look, Dick.
It's got the wings sticking out, right?
Or a brain.
It could be a brain, but
or a very fat hand with a thumb.
Yeah, when I thought about it, I was hungry, I guess.
I've always been very thankful for that all of this stuff that does protein synthesis is actually hung together on a particle that you can isolate.
Yeah.
Okay?
Yes.
Otherwise, we'd be in big trouble. We wouldn't know anything.
No,
This is all true.
I should say that's about the size of poliovirus, these ribosomes.
Yeah.
About 30 nanometers.
Correct.
There's a great quote here by Wayne Hendrickson in the Science article.
He's right here.
Who's right here at Columbia. He's a crystallographer.
Yes, he is.
He says, it's wonderful. Here's how he puts it. It completes the Nobel recognition for the central dogma, so which describes how genetic information in DNA is copied into RNA and then translated into protein. So 1962, Watson, Crick, and Wilkins got the prize for the structure of DNA.
Right.
2006, Kornberg for polymerase, RNA polymerase. And then now we have ribosomes. So the flow of information is complete.
Gentlemen, what's next?
The details?
Here's what I think is next. One day, we're going to learn how to look at that level in a living cell.
Mm-hmm.
And you can then see the true architecture of a cell. It's not a bag of proteins or a soup in which all of this stuff is found. There's microecological relationships between these molecules, as we've learned from studying the membranes, for instance, and how lipids move around and how the bilayers behave. All these molecules have places to go to and have ultrastructural places. I mean, when you do the electron microscope pictures of a cell, you don't get a big feeling for how all these proteins interact with each other.
We're starting to get to that point with things like two-photon microscopy.
Yes, we are. Yeah, listen, that's the point I'm driving at, is that we will go to however far the length takes us in order to see it.
Yes.
In order to see it. Remember Marty Chalfie, who's also a friend of ours, got his Nobel Prize for looking with green fluorescent protein, which you can now use in living systems.
Mm-hmm. Yeah.
So we want to see things. We have to see it to believe it, and it doesn't matter what the level.
I think you're right.
This super collider hadron, the big particles.
Hadrian was an emperor.
How do you look at them? You look at these trails that they leave, right? And you look at all the subsets of particles.
Suppose.
You still look to see what they are. So it's all visual.
So what you're saying is we're going to make a hadron collider for cells.
No.
I'm sorry. I'm just kidding. I understand. But that's going to be a long time. We're going to be all gone, all four of us.
I don't know. Technology is going at a rapid rate.
Not to be morbid. It's going to take a long time. Real-time 3D structure? No, it's going to take a long time.
Well, some electron microscopes can do that now. These very powerful electron
But electron microscopes, you have to fix the sample.
No. They can use living cells in some cases.
Oh, yeah?
Yes.
Well, Dick, what I know is that these guys would like to predict the structure without doing all this crystallography, right?
We knew somebody like that, didn't we? We knew Elvin Cabot.
Yeah, well.
Who worked out all of these rules of protein synthesis.
Well, you know where he is, don't we?
We do.
That's why I'm saying
we don't know all the rules yet. That's the point.
We don't know the rules. But the one rule that we do know is that we need to take a break.
We do.
To acknowledge the support of TWiV by Citrix, the makers of Go to My PC. We do have up and coming a very interesting story on pox viruses, so stay with us, but we need to acknowledge our support. Now, most of us, including you, Dick, use a BlackBerry, an iPhone, a Palm to check your email when you're out of the office.
Yep.
But it's not enough. You need to get your files. You need to get your programs. You need to get your data. So these mobile solutions are great, but you need some more. What's the key? Go to My PC. Complete access to everything on your computer from anywhere in the world. With Go to My PC, you're actually operating, get this, you're operating your office computer from any other computer. Isn't that amazing?
It is amazing.
This program allows you to do that. You can access your programs, your files, everything you need to work from anywhere in the world, and all you need is an internet connection, which is almost ubiquitous, but not quite. Here's what you can do. Our listeners can try Go to My PC free for 30 days, 1 month of unlimited remote access for free. You ought to try it, Dick, and Rich and Alan.
I think I'm going to try this. You're talking me into it.
My only problem is I have an Apple. It's going to be hard to do that with an Apple.
You can run it in virtual PC mode, Dick.
Oh, I never thought of that.
Yeah. For this special offer, though, you got to go to their website, www.gotomypc.com/podcast. One more time, www.gotomypc.com/podcast. Free 30-day trial. And, of course, we thank Citrix for their support of This Week in Virology. Now we have an interesting title of a story called Outfoxing Pox, Developing a New Class of Vaccine Candidates, which was sent to us by Jim from Virginia. And this is an interesting story, which Rich knows all about, I'm sure.
Well, I know something, I think. I've been working on this. So the original article that was brought to our attention was in eScience News, and it refers to a virology paper about trying to develop a new smallpox vaccine. So just as background, let's recall that the original problem here is smallpox, which was a very nasty disease that was eradicated by a worldwide vaccination program that used a live vaccine called Vaccinia virus that's many people are familiar with. It gets scratched into your arm and gives a little blistering lesion and gives you immunity to smallpox. It's a very complex virus, as viruses go. Double-stranded DNA virus that encodes about 200 genes. So we're looking at a lot of gene products, and that complicates the whole issue. After the disease was eradicated in late 1970s, we quit vaccinating against smallpox because there are complications associated with the vaccine, and the risk of dying from the vaccine was greater than the risk of dying from the disease. And so everybody just kind of forgot about it for a long time.
Right.
But then we had 9/11, and shortly after 9/11, some loony mailed a bunch of anthrax around, and everybody woke up and said, whoa, there could be a bioterrorist event.
Yep.
And our government responded by pumping a bunch of money into biodefense research, and the top of everybody's pick list for a bioterrorist agent was smallpox. And so this stimulated a lot of poxvirus research. In particular, let's look at vaccines and anti-poxviral drugs. And so there's been a lot of research on poxvirus vaccines over the last 8 or 9 years.
Right, because the previous vaccine essentially was what Jenner had used. This was an ancient thing, right?
Essentially, that's correct. And there are side effects associated with it. There's about a 1 in a million chance you'll die from the vaccine, and there's about a 1 in 100,000 chance that you'll have a serious complication. And these include mostly in immunocompromised individuals, and there's a lot of those around nowadays, or people with skin diseases. You can get the vaccination spreading on the skin, or you can get progressive vaccinia, and these can be pretty nasty conditions. So there are complications associated with the disease, so there's motivation to come up with better vaccines. This paper is really interesting because it really raises the issue of the difference between live vaccines and either subunit vaccines or killed vaccines, the advantages and disadvantages of them, and the difficulty associated with creating a vaccine that's really effective. So the live vaccine is great. It works really well, but there are side effects associated with it. So a lot of the research that's been stimulated has to do with alternate vaccines, either more crippled live vaccines or subunit vaccines or killed vaccines. So this paper deals with generating an appropriate subunit vaccine. And by that, what we mean is take some protein that's made by the virus and somehow inject that or introduce that into people so that they mount an immune response to that protein and hope that that immune response then protects you against the virus infection. One of the big questions is, with something like a poxvirus with 200 genes, what do you pick, right? So there's some background information that you can go on. People have looked at what the normal immune response to the virus is, either looking at T cell epitopes or B cell epitopes. So for those who are not into this, there are two arms of the adaptive immune response. One is making antibodies, and the other is making T cells, special kinds of immune cells that react with a specific target on a cell, a virus-infected cell, and kill that cell. And you can, in a person who's experienced the disease, you can look and see what their response has been to the infection, either in terms of the antibodies they make or the immune cells that they make. And people have done this for poxviruses and looked at what are the antibody responses, what are the T cell responses, and there's a whole bunch of them. There's like 75 different 75 different genes that make T cell epitopes in the virus. That's almost half of them. There's at least 35 different genes that you mount, you make antibodies to. So if you're going to go make a subunit vaccine, are any of those better than any of the others? Do you have to do all of them? You still have a complex problem. Another approach is to use candidate genes. Okay, so obviously surface proteins are a pretty good idea, and this virus comes in two different forms. One is the basic enveloped form, and the other one has a second envelope. So you got two different surfaces to deal with. The most basic envelope form has 20 proteins on the surface. The other one has another 8 proteins. So now you've still got 30 proteins to choose from. Well, people have sorted through this and tried to figure out the best. And one of the best that was come up with was a group at USAMRIID that concocted a vaccine that they call 4-pox that has 2 proteins from the outer envelope and 2 proteins from the inner envelope. And they delivered this as a DNA vaccine. So you just make plasmids from these and use, I'd never encountered this word before, biolistic
That's a new one to me too.
Yeah, I had seen that. That's been very popular in plant research, and I always envied those people because they actually get to shoot their cells.
Oh, is that what it's about? Shooting?
In fact, some of them operate on a 22 blank cartridge. I don't think the one for medical use uses that, but you actually load the gun and you fire this thing at your cells.
So I'd been aware of the technique, but I'd never heard of the term biolistic. I like that. Sort of a pun on ballistic, I guess. You take apparently gold particles and coat them with DNA and then shoot them into the skin. Okay?
And maybe we could use that for our episode title in some way. Biolistic, right?
There you go.
Do they do this in mice?
Yes, in the experimental system as mice, you shoot them in the ear. Okay?
And this is with DNA, right? With plasmids.
With DNA. Wow. So you introduce the DNA. It's got appropriate promoters on it and that kind of stuff. It expresses a protein. And so that's how you indirectly get the protein into cells. And so the cells have a chance to mount an immune response. So this 4-pox in at least one mouse model was fairly reasonable. Gave not as good as a live vaccine, but in one mouse model, pretty good protection. So this group, sorry for the long introduction, but it's good background. This group said, okay, we want to take a different approach to identifying what the appropriate antigens are. And it's really a heroic effort and pretty clever. What they did was to clone every gene from cowpox and then test these genes delivered as a DNA in a DNA vaccine in pools. To mice, and then ask whether the mice are protected. In terms of detail, we don't have to go into all the details, but it's really there's a lot of technical stuff that they did that's very interesting. They actually took all the genes and broke them up into 500 base pair open reading frames because they wanted them all expressed at the same level. They did codon optimized them so
very cool. Yeah, that was very cool.
Did stuff as they actually expressed them in pools rather than doing individually, and then they created 75 different pools, each with about 30 open reading frames in them, in 3 different groups, so that each gene was represented once in each of these 75 pools, but in a different context. So it was sort of internally controlled.
Beautiful.
And they infected mice with all these and looked for. I really like this part. They didn't fool around. They didn't look for antibodies or T cells or something like this. They
protection,
Yeah.
Okay. They went right for the throat.
That's great. They challenged them with cowpox, right?
They challenged them with cowpox. Interestingly, now they used a different mouse model than the 4-pox was done in, and one of their initial observations was that in this mouse model, and I guess it had been done in spelled C before, and these are C57 black, or maybe it's the other way around.
No, you got it. You're right. Yeah.
Okay. In the C57 black, the 4-pox doesn't work. Okay, so that's one of their points, is you got to be careful with your models. As you've said many times, Vince, mice are not people, right? And even mice are not mice.
Okay? Well, inbred strains are certainly not mice, that's for sure.
You're right. Mice are not mice. Yeah.
Mice lie and monkeys exaggerate. Yeah.
Wait, say that again, Alan.
Mice lie and monkeys exaggerate.
That could be an episode title.
Yeah, that's good.
Wow.
Okay, so when they did all this and sorted through all this, they came up with 9 genes that gave some protection. It's not striking, but it's some. And then they tried combining all those together and see what they get. Frankly, in the end, you know, I'm a little disappointed, and it's not their fault. This is just the way it worked out. It works better in the new mouse model than 4-pox, so that's good. It's not nearly as good as the live vaccine.
Wow. That's too bad.
It's a terrific study in how to do this. It really emphasizes the difference between a live and a subunit vaccine and the complexity in trying to figure all this out. I think it's a heroic study, and it teaches us that we still don't know a whole lot, and it's pretty complicated. It's interesting. You can compare the B-cell epitopes and the T-cell epitopes and what these guys found, and they are partially overlapping sets. You don't have any common denominator in any of it. There's membrane proteins, there's core proteins, there's things on the surface, there are things that are intracellular proteins.
So they, after they used this approach to identify the proteins, they took the whole reading frame for each of the, 6 proteins and immunized? Nine, and,
yes.
They did it separately and all together. That's what you're saying, right? And even at that didn't work as well as the infectious virus vaccine.
That's correct.
It's amazing. It is disappointing because this is a huge effort, and I think this is supported by biodefense money, right?
Yeah.
So
I think so.
The kind of stuff you can do. We could never do this by an R01 mechanism, an investigator-initiated.
It really is. It really is a huge effort. I didn't actually look at the acknowledgments to look where the funding came from.
It's an NIH U54, which is, I think...
That's a project. Yeah, that's probably biodefense money. Yeah.
That's really cool. I think a lot of... You learn things from this. This is like making Teflon, right, for spaceships. You can put it in frying pans. So this technology probably... The whole approach is fascinating.
The approach is fascinating.
It's very
I think just the technological advances that they made in putting all this together are worth the effort.
Cool. I like that. What do they do next, Rich? They have this kind of week.
That's what I was going to ask.
What do they do?
I really don't know.
They don't get their grant renewed, right?
I don't know where you go from this because, as I said, if you compare this, I don't have a lot of imagination when it comes to this kind of stuff, but when you compare this with, it is better than 4-pox. But it's still not anywhere near the live vaccine. But you compare this and what you know about the T-cell epitopes and what you know about the B-cell epitopes and the 4-pox, and I don't see a common denominator. It's not obvious to me where.
That's right.
So, Rich, these are delivered as DNA vaccines, right? Maybe that's not the way to go. Maybe you have to make the proteins or something.
That's a good point.
Right.
Maybe the actual delivery could be optimized, and it would work.
So many variables here.
Yeah, I mean, you could put it into an infectious virus vaccine, another vector. So I guess there are lots of things they can do.
And then there's the question about the strain of mouse that they picked. That's an inbred strain, of course, and the other one that you mentioned was also an inbred strain. And I now need to tell you about an anecdote. I won't take much time with this, but I worked with Racaniello, which is a very large parasite and which has many, many antigens of various kinds. But if you take, I think they were up to like 35 different inbred strains of mice, and if you gave each mouse strain, these were all 3-month-old male mice, you gave them all the same number of infectious units to begin with. In this case, they're the number of larvae that you give orally, 200 per mouse. Every single inbred strain of mouse will end up with a different number in their muscles. Every single one of them. There isn't 2 alike. And you can line them all up, and you can say the DBA2 mouse is the most resistant and the C3HEJ mouse is the most susceptible, and you can show all the intergrades in between. So if I were doing vaccine work, and I happen to pick the wrong strain of mouse to begin with, I'm going to get a result that you won't be able to talk about.
Well, you know, in a company, you do many different kinds. You do many strains. You do many animals, and you just hope when you go to people, but it sometimes fails.
When you go to people, we're not.
You know, you can have drugs or vaccines that work beautifully in animals, and you put it in people, and there's no effect, and that's the end of the line.
Very common.
No kidding. So it's a miracle when something does work.
Yeah, we should cut some slack to the pharmaceutical companies. You know, I know they want to make money, but they also do make things that help people.
Yeah.
Right.
Right.
Yep.
All right, that's great. Thanks very much.
So is the bottom virology line on this still that live vaccines are better than killed vaccines?
Well, there's no such thing as a live vaccine.
I mean, an attenuated virus that replicates.
So far.
An attenuated replicative viral unit.
I would say, me personally, I would say that infectious vaccines, attenuated ones, are still better than non-infectious.
You start with
ones.
the gold standard of yellow fever virus vaccine.
You know,
right?
so the flu mist, the infectious flu, is a little better than polio. Right. Yeah, but that's not going to be forever. As soon as we get good at delivering the right things to the right places, it'll change, right?
And the other question was, even if this had been successful, how long would the immunity last?
Yeah, those are perfectly good questions.
Right.
I presume 4-pox never has gone into people, Rich, right?
I don't think so.
Yeah.
Oh, I don't know. Okay, I have no information on that. I don't know.
Well, you know, if it's not good enough in mice, it's not going to go into people, right? Because it's rare that it gets better in people than in a mouse, although that could happen. But it's hard to justify because people's studies are expensive, right?
Pretty sure.
All right. Let us... Hey, Alan, should we do this Canadian story? I think we could
we might as well, eh?
Yay. Okay.
So this will be fairly quick. There's the Canucks are all in a tizzy about...
Alan, you better start again.
Sorry.
just go right back to the beginning, please.
There's been quite a lot of media coverage in Canada.
Thank you.
Did he just say the same thing? Was that?
Yes, I think I did.
Okay.
It sounded the same to me.
I could repeat it in French if you like.
No, that'll.
No. So there was a study that was done by public health officials in British Columbia, which is in Canada.
Indeed.
And they looked at rates of H1N1 infection in a population, and very few people have seen these data because this hasn't been published yet. It has reportedly been submitted to a journal. We don't know what journal. The lead authors are very appropriately keeping their mouths shut about it, but unfortunately, other people who have seen the data are not keeping their mouths shut about it. So supposedly, the results show some correlation between having gotten the seasonal flu vaccine last year and catching H1N1 this year.
So if you got the seasonal vaccine, you're more likely to be infected with pandemic H1N1.
Right.
Yeah.
Is the suggestion reportedly secondhand from this unpublished study? Well, because this leaked out, and shame on whoever leaked this out, because that's what the peer review process is for, to figure out if this is valid and worthy of publication. But now it's gotten all over the Canadian newspapers, and this has had a predictable effect on people's desire to get the seasonal flu vaccine in Canada, where, of course, it's flu season. So there's, all over there are these follow-up stories about it and what's going on, and they're browbeating these people to release the data, and they're trying to figure out, what exactly does this show. Meanwhile, the CDC and the WHO have said that they're looking at other data which are actually available. You don't see this effect. In fact, the CDC data from the United States show no linkage between no increase or decrease in risk of getting H1N1 based on whether you got last year's flu vaccine, seasonal flu vaccine. There was one small study in Mexico that was published that found the seasonal vaccine might have provided some protection against the H1N1 this year. So the data that are available that have actually been peer-reviewed and published suggest there's either no linkage or certainly no linkage you need to worry about. this unpublished study from an unknown number of patients with unknown controls. We don't know, but this is the type of thing that happens when somebody decides to go ahead and go public with a partial bit of information that they think they understand.
Alan, there's two things wrong here. One is that they released it to the press, and secondly, the press produced it. The press shouldn't have taken the article.
I can't really blame. I don't think you can really blame the press for putting out a story that they've been given.
Come on, they have a right to refuse it if they don't see enough evidence.
It's been reported. I mean, the news reports about it are straight to the facts that they have available. They say, sources say that this is what this says.
It's a rumor mill.
And they quote other folks saying it's published. Yes, this is an incendiary story, but that's what sells newspapers.
I normally wouldn't discuss this because we can't see the data.
Right.
And in fact, if these data ever come out, we'll talk about it. But there's a bigger issue here, and that is that stuff that's being reviewed shouldn't be leaked. There was actually an editorial in one of these Canadian papers where they said we should see all the data before something is peer-reviewed. But that's wrong, in my view, anyway. I'd like to hear what you guys think. I mean, as you said before, Alan, the peer review process is to make sure that whatever a conclusion of a study is made, it's valid scientifically, right? So I agree.
I think the peer review needs to come first. My question about this is that the dates on these are a month ago.
Mm-hmm.
Yeah.
Okay?
Yeah, this has been going on.
I can review a paper in an afternoon.
Right.
Yeah.
So what's going on? Do you know anything about this, Alan? Is this
I
just
don't. I haven't
a month
seen,
ago and we haven't heard anything since?
I have not seen it coming. I mean, I wouldn't be able to tell you if I'd seen the embargoed paper. I wouldn't be able to tell you what was in it. But I can tell you that I have not seen the embargoed paper. It hasn't. Not that I see all of them. But but in my news feeds and such, I have not come across this. I don't see any follow-up stories saying that it's been published. So I don't know what the holdup is in this day and age of electronic publishing and electronic transfer and peer review. As you say, Rich, it should take an afternoon for the peer reviewers to go through this thing. And given the urgency, I think they really ought to get on the stick.
Yeah, I would think in a situation like this where, okay, yeah, you should do the peer review first. It shouldn't be leaked, all that kind of stuff. But something got leaked.
That horse is out of the barn.
So now we have a situation, and under those circumstances, I think is the responsibility of the scientific community to clean this up as soon as they can. And the way you do that here is to complete the peer review. Now, it could be that what's going on is that the peer reviewers said,
Yeah,
right.
Yeah, sure.
This is no good.
That's right.
So then what do you do?
Well, maybe they asked for more data, and they're doing
Right.
that right now. In which case, it's still in peer review, so they can't release it. But, guys, you've seen all these hot flu papers. They come out in a week. They're reviewed in a day.
Right.
And Nature and Science, they publish them within a week. So you're absolutely right. This can be done. So I suspect there's something wrong with it, but we don't know. But
let's hope that it's not just a hoax.
Under those circumstances, would you, if the peer review says there's something wrong, ordinarily in peer review, somebody says there's something wrong, you send it back, you got to do more experiments. That may take time. In a situation like this, then do you make some sort of pronouncement that it failed the peer review and they're looking for more data? That doesn't seem right either. I don't know what you do.
I think what the editorial was arguing, I mean, Vince, I partially agree with you that you don't just bypass peer review because part of the paper leaked out, but I think the underlying argument there is, look, enough of this has leaked out to do harm. So the quickest way to diffuse it is to just let the whole thing go. Say, okay, here are the data. No, it's not publishable quality yet. And if there's some outstanding issue in the peer review, do that as a transparent process.
Yeah, but Alan, I don't think people could understand. A lot of people wouldn't understand the data.
I think, of course, a lot of people won't understand the data, but a lot of people will, and it would give a whole lot more credibility to discussions like the one we're having right now, and what the CDC is saying and the WHO is saying, where they would be able to point to this and say, our data based on X number of thousands of cases indicates this, and this small data set from Canada, which has flaws A, B, C, D, and E, it says this, and therefore, we're not putting a whole lot of credence in this Canadian study. Or,
Sure.
The alternative is maybe the Canadian study does have something to say. I don't know.
Yeah, I think in the end, this story doesn't help anyone, because if this paper's rejected and it's leaked, then people are going to say, "Wait a minute, why was it rejected? How do we know that they're not hiding something?" Everyone, they're going to be suspicion, distrust, all the things you don't need for vaccines. So
yeah, I think because of the exceptional circumstances in this case, it would actually make sense to just release the data.
Yeah.
But I would ordinarily agree with you, Vince, that this would not be the way to go.
Yeah.
There's something very important, I think, for the listeners to understand, and that is that none of the peer-reviewed published studies support the conclusion that the seasonal vaccine increases your susceptibility to
That's right.
H1N1.
That's right. We do actually have data on this that argue that this is not a problem at all.
Right.
So,
with influenza, that's correct. Right. There are other viruses where there may be some issues, but I agree with influenza. Unfortunately, it's seasonal, which I would say if there's anything here that it's not so bad that people don't want to get a seasonal vaccine, because it's probably not going anywhere. But,
and, I think we should point out that we here at TWiV put our money where our mouths are. I got my flu shot.
You got your seasonal shot?
Got my seasonal shot.
Right.
I missed the local free flu clinic. I had to shell out $30 for it, but I figured it was worth it.
How about you, Rich?
I got mine. I'm waiting for the H1N1.
Yeah.
So here we at the medical center, we
the outlier so far.
We haven't gotten our H1N1 here yet. They're still waiting. We have 23,000 employees. 18,000 of them got the seasonal vaccine. But and this is an at-risk population, as you know.
Absolutely.
So it's unusual that we haven't got. By the way, there is a CDC page now where they tell you on a weekly basis how many doses of vaccine go to each state in the U.S. They're being very transparent about that.
Yeah, they're having a lot of supply problems this year.
Yeah, not surprising. All right, our last story is a quick one, very interesting on ProMed Mail. A guy in what state? Oregon. He had a ferret. He got flu, and then the ferret got sick. He brought the ferret to the vet, and the vet was smart enough to have it tested, and it turned out it was H1N1. So the guy had H1N1 flu, and then he gave it to the ferret, which is the first documented case of a ferret getting it from a human. And,
unintentionally.
Unintentionally.
Right.
So this is a good point, Rich. All right, assuming he doesn't kiss his ferret.
He may.
He was.
Don't make that assumption.
Yeah, I'm assuming. But I understand. This is Oregon, right?
Ferrets are cute.
Ferrets go all over you.
If you're in the same room, you're making an aerosol, right?
You are.
There you go. And if he maybe he had the virus on his hands, and he, took the ferret out of the cage. So it just kind of underscores the transmission issues at play here. But ferrets.
And I'm glad to see in the story the ferret is recovering.
The ferret is recovering.
A
yes, as you know, we've talked about experimental infection of ferrets with influenza virus. They're highly susceptible. So, in the wild. Not the wild, it's a pet ferret, but this just confirmed. So I thought it's a great story. We'll put a link to that.
Right.
Let's do a couple of emails. So Dick is getting antsy here, as usual.
No, he's not.
No? All right.
I just have my pick of the week. I'm anxious to tell you about it.
All right, we have a long one. I'm going to skip that one. We'll do that another time when Rich is back.
Yeah, I want to be here for that one.
We'll have it here, but I think it would take us a while, so I want to do some short ones. We have one from Ariel. Hello, Vincent, Dick, and Alan. I very enjoy listening to your podcast. It's one I never miss. I'm very fascinated about viruses since I was a child. Today, I teach biology, science, and technology in a high school in Israel. In the past, I learned some virology when I tried to get an MSc in virology at microbiology faculty. You tried? You didn't get it? I recently read a summary article about epigenetics, and I was very interested. Did someone try to figure it out if epigenetics play a role in viruses? Does epigenetics exist in viruses? Thanks. Well, I think so. I mean, methylation of genomes is an epigenetic way to control viral gene expression.
Yeah, absolutely.
Yeah,
Absolutely. Any modification of the DNA that's not a DNA sequence change that's going to be heritable. VPG and poliovirus pops to mind.
What about you, Rich? Any other ones? You got? You work on a DNA virus. That's a good one.
Yeah, I can't. Hard for me to. Well, I mean, I'm cheating here. MicroRNAs, I think, is a good idea.
Sure.
Okay. I don't know if that's really epigenetic. Would you call that epigenetic? Because microRNAs are kind of gene products in their own right. Okay.
I suppose, yeah.
Yeah.
Yeah, you're right.
There's no RNA editing going on here in RNA viruses.
Oh, no, that's a good point.
Yeah.
Would you call that epigenetic?
Yeah, I would guess
'Cause
so.
there's plenty of that going on.
It's not encoded. The reason I thought microRNA is because they're not encoded by the viral genome. So.
Okay.
And it's
so
not
you got
inherent. It's not
cellular
inherent.
microRNAs. I was thinking
Yeah.
of microRNAs encoded by the
Yeah,
virus.
cellular, and they're not inherited in the viral genome. So that could be it. Yeah. So there are many. Yes. So the answer is yes. We have epigenetics. We talked about that way long time ago. Remember?
We did.
Because we were talking about genetic mutations, and someone said genetic mutations, probably the New York Times, and I objected to that because
That's
there's no other kind of mutation.
Exactly.
So some listener said, "What about epigenetic?" I said, "Well, then it's not genetic, right?" So,
Right.
Okay, let's see here. We're going to do this next one is too long. We'll skip that one, and we'll do the last one, and then get to our picks. This is from Ben. Hi, I am not a scientist, researcher, or health professional, just a closet science nerd who loves your show. I just had a few questions about viruses that I can't seem to find answers for that I was hoping you could answer. Are there virus receptors on cells themselves? If so, why would a cell evolve a receptor to something harmful? How did these viruses evolve in general if they kill off their hosts? That certainly couldn't be a beneficial trait, right? So I think we could deal with that first, right? So
Right.
there are virus receptors on cells.
Of course, how else would the virus get in?
But they're not there for the virus.
No, that's the other thing to say.
So they have cell functions, and viruses have evolved.
They have taken advantage
To
of
take
existing
advantage of them,
systems.
Right. How do viruses evolve if they kill their hosts? That's not a good trait. Anybody else want to deal with that one?
Well, I would say he's absolutely right there. I mean, you got to killing the cells is part of the reproductive cycle. Okay, so that's in most cases you kind of have to do that because as a virus you're changing everything so much the cell's going to die. But it's in the long run. No, not necessarily a beneficial trait. The most successful viruses can persist for a long period of time, don't necessarily kill the host or the cells.
Yeah. So there are lots of viruses that persist, right? But even the ones that kill cells, they don't kill all their hosts, right?
No.
Right.
There's always some left. So
There are
And
some
the ones
notable
that are,
exceptions.
The ones that are successful manage to kill their hosts slowly enough that the hosts can pass the particle on.
right. I guess
Well-
a good example are Ebola, hemorrhagic fever viruses, which are quite lethal. They don't spread very much, but they're fine in their natural reservoir. So
Right,
maybe,
they're quite lethal to humans, and they're also not terribly successful in humans if you measure that as number of infected hosts.
so that's a good point. So there are hosts for many viruses where the infection is not lethal. So the hemorrhagic fever viruses are lethal in people, but in bats, they persist without apparently harming the bats, right?
Right.
Yeah, and I was going to say rabies is another one of those that we're an accidental host.
That's right. So we're not important in maintaining those viruses, right?
Precisely.
So
And as you pointed out before, and the classic example of this is the introduction of myxoma virus into Australia.
All right.
It starts out as a very lethal infection, and
Yep.
then over time, an equilibrium is established where the rabbits become more resistant and the virus becomes less virulent. And that's really the natural evolution of things to the point where you reach an equilibrium where the virus really doesn't do a whole lot to the host. It just sort of coexists.
Yeah. Using people, when you think of people and viruses, it's misleading because they're often accidental. But, even XMRV is in a lot of people, apparently, which we talked about today, and they're fine.
Sure.
So, okay, then his next question is, how are viruses so infectious and damaging when they are merely an envelope of genes? Since they aren't alive like bacteria, I can't imagine why they would exist just to kill things. Well, we answered just that right now, right?
Yep.
The same goes with prions. There can't seriously be an infectious protein, can there? Proteins are just amino acid folds. Hope you can answer some of these either by email on your show. Thanks a bunch. How can viruses be so infectious? Well, that's why, because they're a package of all the right genes, right?
Yep.
Rich?
Yeah, so as we said before, the whole idea is to go in and take over the cell, for the purpose of replicating this particular nucleic acid, and the cell suffers as a consequence, and that's where the infectivity and the killing comes from.
Yeah.
As for prions, boy, I'll tell you, there were a lot of people who were very skeptical about the infectious protein concept there to start with, but it's very well established that these proteins interact with normal proteins and change their conformation into bad proteins, and that's propagated. And in a sense, that's an infection.
And really, any mechanism that can propagate your genes to a subsequent generation is a potentially valid evolutionary strategy. It doesn't have to be nice.
Yep. No, I think the prion, Ben, we're going to do a show on prions, and we'll get more into that. But Rich is right; they can propagate themselves in unusual ways. My view of this bag of genes is that these are what have evolved the minimum to take over a cell, as Rich has said. They're very efficient.
But why haven't I as the other non-virologist here, besides Ben, I could ask the same question in another way. Why can't a virus sneak into a cell, replicate itself, and sneak back out without being detected?
Many do.
And don't kill the cell.
Many do.
Oh, well, then Ben should hear that then.
So many do get in a cell and don't kill it. We have some we have these polyoma viruses that don't kill us, right? And in animals have lots of viruses that don't kill them.
So they don't actually take over the cell. They actually depend on an intact cell to reproduce themselves without getting caught.
Yeah. I mean, the point is you hear about the lethal infections, but all the ones that are not killing you don't hear about, right?
Yeah.
And I would wager that in the globe, well, this is wrong because of the ocean, but most of the infections are probably not lethal. The problem is in the ocean, there's huge turnover of bacteria by bacteriophages. They actually kill them, and that's important for the carbon cycle. But on land, terrestrial viruses, I would bet most of them are not lethal to their hosts.
What's not clear to me is whether there are good examples, maybe you can help me with this, good examples of cells that when they're infected are really doing exactly the same thing as they would be if they're not infected.
Right.
I mean, usually the virus, even in a persistent state, affects the metabolism in the cell. So it may not carry out its normal function properly. It's become sort of a home for the virus rather than what it was originally doing.
Well, it depends how you define infected. I mean, a lysogenic bacteriophage.
Yeah, that's doing its normal job. And in fact, a neuron that's harboring a herpes virus
that's
probably functioning okay as a neuron.
Sure.
Yep. There you go. And all these endogenous retroviruses, right? They're there.
Right.
And they're not harming. So lots of examples.
That's right.
Okay, Ben, and maybe that clears it up.
Yep.
All right, guys.
I want Ben to know that these are great questions.
Yes.
They're wonderful questions.
No, that's beautiful. It's really good. And you are a closet science nerd.
Absolutely.
I hate to say it, Ben, but you've just come out of the closet.
Yeah.
But being a nerd is good. It's good stuff.
Nerd is good.
Yep.
All right, let's go on to our picks of the week. We've heard from Jason, which was great.
Yes.
And since, Dick, I see your face there.
I have a wonderful pick.
What's your pick?
Well, if you go online to this month's Scientific American, there are two wonderful things in there. One of them is very self-serving. I have an article on vertical farming. You can forget that because I don't want to really toot my own horn here. But
it's a whole issue on the sustainable future.
That's right. But they have published the Nikon Small World Contest winners.
Ooh, goody.
And they are fabulous. They are.
Ooh, I'm going to look at that.
They're absolutely fabulous.
I'm sorry, Dick. I'm looking at your article.
Don't do that.
Nice.
But it's okay. I mean,
by
it's fine.
Despommier.
It's fine.
How did you get to write an article in Scientific? All right, but
they asked me to.
The Nikon winners. What's that? Where do you find that?
That's on the same homepage. Go back, and then they list them down at the bottom, as I recall, because I was looking at them this morning.
I just did the search. I got petite pictures.
That's the one.
20 microscopic photo competition prize winner.
There you go.
Great.
Okay. Alan wins the best Google search. He's very quick.
Really quick.
Nice. Very nice. Photomicrograph.
They
I
are
dig up information for a living.
They're fabulous. Yes, you do.
Yeah,
Yes,
he
you do.
does.
Well, you had a hint. Come
Are
on,
you
you had a
sitting in
hint.
front of 1 or 2 computers?
2, but we're only using 1.
I
you mean Alan?
Nice.
I have
Oh,
two
these are
screens on one of them.
beautiful.
They're fabulous pictures,
Look at
guys.
this.
They're visual. Remember, we're
What did you
visual
Google,
animals.
Alan? I can't wait for this link.
Oh, I didn't Google it. I
Just,
searched the
yeah.
SciAm site. Let me. I'm pasting it into the show notes. If you're looking at the show notes.
Look at these things.
These
I
are
mean,
beautiful.
they're fabulous.
Very nice. These are gorgeous. Oh,
There's my
man.
spiral.
These are gorgeous. Starfish. Wow. Beautiful. That's nice, Dick.
Yep. So
Have you found it yet, Rich?
No.
Go to the Scientific American site and just search for petite.
Okay.
that
I'll
looks
get
like a
there.
fungus of some
That's
sort.
very fungus. Yeah, that's beautiful, Dick. And what's this?
Nanotubes. Got to be nanotubes.
Nice. Cotton fibers.
All
All right, that's
those are
great.
non-nanotubes.
All right, well, that's a good one, Dick. Very nice. Thank you.
Glad
very
you guys
much.
liked
Plus,
it.
your article is very nice.
That's fine.
I got it.
Okay,
Got it,
okay.
All right. Aren't they beautiful? Well, you look through them while Alan gives us his pick.
So my pick, I actually had to go back and search through the previous agendas because I thought, gosh, I must have mentioned this before, and probably a lot of listeners have already heard of it. It's Make Magazine and the associated blog. And if you have not already found this and you're listening to TWiV, well, you need to find this. This is a magazine that started up a few years ago. It comes out quarterly, and the blog gives you kind of a flavor of what goes on in the magazine. It's all about do-it-yourself projects with as broad a scope to that as possible. It ranges from
Really?
amateur chemistry experiments that you can do in your kitchen or garage to building microcontroller-operated props for Halloween. Just absolutely every type of thing that you could imagine.
Vertical farms.
Or maybe not imagine
A
doing
Mr.
yourself.
A Mr. Wizard manual.
Exactly. It's a modern version of that.
I love it. I love
It's
it.
a better way to slice a pumpkin.
Yeah. A better way to slice it.
Yeah, this is great stuff. And they also have make fests, right, where you go
they
and
have-
they have
You think
people
this is
doing
mostly a guy type magazine?
No, it's not. The editors are women too.
Are they?
Absolutely
Okay.
not guy.
Yeah, it's a mixed
Okay.
group.
And they have fests where you can go and watch people do this stuff. It's very cool.
Like it.
That's beautiful. No, I think a lot of our listeners would probably already know. We have some technology listeners, but for those who don't, this is very interesting. Do
Parts
they
you
actually
can
have?
salvage from an old house.
Do they ever have a? Do they have a paper magazine, or is this online?
Well, there's
only?
a paper magazine. It's about $35 a year to subscribe. It's a quarterly thing. Very, very nicely produced. And I get it. It's a lot of fun.
Beautiful. Thanks, Alan. Hey, Rich, have you looked at those pictures? You like them?
I haven't heard anything you guys have said. Because I'm looking at these pictures.
They're pretty nice.
They're amazing.
Rich, I love it. I love
Yeah,
it.
it's good.
Just amazing.
Yeah.
You,
Okay, so is it my turn?
Yes.
Yes.
Okay, so I looked back at past episodes as well because I couldn't believe that this wasn't already there. All you got to do is watch a lot of TV for this one. I'm going to recommend the BBC series Planet Earth.
Ah, it's the best.
It's really good.
It is the best.
There are 11 50-minute episodes.
You're right, Rich. You're absolutely right.
And
They're
it's
fabulous.
all about it's kind of like what was the one that they did years ago? Life on Earth.
Yes.
And as a matter of fact, the British edition of this is narrated by David Attenborough.
That's right.
The American edition is narrated by Sigourney Weaver.
Weaver, exactly.
I listened to the British edition, and they do it. They go through the planet, a lot of photography. It's a nature show, biome by biome.
Here, here.
They do caves. They do deep ocean. They
Caves
do
are great.
seas. They do the ice world, etc.
Yeah.
And the emphasis really is on relationships.
Yep.
And ecology and the very delicate balance
Exactly.
in nature. And
Exactly.
it's just absolutely beautiful. But if you watch this, you got to promise me that you'll get all of the discs and watch the last one as well, which is Planet Earth, The Future. And that's about 4 episodes, I think, 3 or 4, where they interview the makers of the film
All right.
and ask them, okay, what next? Where are we going? What's going to happen? And of course, the emphasis here is on how humans are upsetting the balance.
Yeah, sure. Absolutely.
And what are we going to do about that? And
How about that?
It's good because lots of times you come out of that feeling like, we've had it.
That's right.
And this has a nice balance. Yes, there's some bad stuff going on, but the people that they interview have some optimistic ideas, and that's nice.
Sure.
So it's a great series.
What's your favorite segment, Rich?
Oh, wow.
Because I have one.
I can tell you my favorite moment in the whole thing, and I forget quite what segment this was. It may have been Shallow Seas. I don't know. But my favorite moment in the whole thing was they went out and filmed great white sharks hunting for seals.
That's right.
Now, the seals hang out in a certain area, and then in order to get their meal, they got to go out some distance from their home and go fishing.
Yes.
And the sharks know this, and they hang around in this place where the seals have to run the gauntlet of sharks.
Yeah.
Okay? And they got this amazing picture of a great white shark completely clearing the water with a seal hanging halfway out of his mouth.
Exactly.
And I'm just in awe of this thing. That is nature. Okay?
That's right.
It's brutal.
Here.
It's really brutal.
Yeah.
That's the way it is. It's amazing.
Cool.
That's a human term, brutal.
Yes, exactly.
Nature red in tooth and claw. Yes.
Yeah. Well, my favorite one is when they were looking for the snow leopard, and they were way across the valley. They have these special cameras now, and they can hone in from 2 miles away without disturbing the wildlife.
Right.
And you saw that one, I presume, where the snow leopard was chasing the something like a mountain goat down the slopes, but the slopes were like at 90 degrees to the surface of the land. I don't know how they were running down this thing, but the snow leopard hooked it with one claw, and the baby animal was struggling, and it got loose and fell into the river and went downstream and got saved.
Yeah. There's a terrific sequence too about all of the different fungi that parasitize the insects in the jungle.
Yeah. Oh, that was
very
a great one.
species specific.
That was fabulous.
That's all really alien stuff.
Yeah.
It's really good.
Yep.
You know, we could make one about viruses.
We could do that.
Should
be just as
and it would be right.
And the last one could be the future.
Well,
I'd love to do that one day. That's great, beautiful. I like this pick stuff because I'm sure the listeners like it. I've had many people say they love doing stuff we recommend, but I haven't heard of most of this stuff.
And by the way, Rich, Planet Earth is now a book also, so you can get the book as well
great.
as the. I got the book, and I got my son all the 9 discs for him to look at. He loves it.
Well, my pick is another magazines. It looks like we have a magazine pick day today, 3 of them. Mine is the November issue of Wired magazine, and it has a really nice article on vaccines. It's called An Epidemic of Fear, How Panicked Parents Skipping Shots Endangers Us All. Cover story, little baby with the word fear in big letters.
Sounds like the voice of reason.
And it's very well written. It's good, simple stuff, and it has a lot of interview with Paul Offit, who is a pro-vaccine guy. So check that out. Yep. And also it's accompanied by a page called The Misinformants, Prominent Voices in the Anti-Vaccine Crusade. All these famous people who are speaking out against vaccines. Unconscionable. I heard today that a comedian, now you guys all know about Twitter, and you can have a lot of people following you. So there's a comedian, I forgot his name. He's got 60,000 followers, and he tweeted that you have to be insane or an idiot to get the flu vaccine this year. Now, what does this guy know about it that he's proclaiming to people who are listening to him and probably hanging on his every word? So
he knows that he'll get publicity by saying outrageous stuff like this.
Yeah, it's about outrage, sure. But it's really disturbing to me, just as a virologist, because it's totally wrong, right?
Yeah.
Anyway, so this has a list of people, the misinformants. So not all of them, but some of them, very famous people. Okay, that should do it for another TWiV. And if you have just listened to TWiV the first time, Go over to iTunes and subscribe. You can leave some comments. That always helps us. If you can't use iTunes, go ahead to TWiV.tv and you can download episodes. I now put a player on TWiV, so you can actually play the podcast right on the website. And there we have show notes as well. And TWiV is also part of sciencepodcasters.org and promednetwork.com websites where you can find high-quality science podcasts in a wide variety of fields. Dick, thank you.
Vince, it's a pleasure as usual.
Dick can be found at
Racaniello.org.
Racaniello.org. Rich, thank you.
Thank you. I love this. It's great fun.
And Rich is at a website which is too long to mention. We'll have in the show notes. But Rich, you will be back next month, I presume?
I hope so.
You
yeah,
shall.
I'm all for it.
Good.
So Rich and I are trying to arrange a visit for me in Florida for a seminar, and then we'll do a TWiV. We'll do a TWiV.
Sounds like a plan.
From Florida. Right. And I'll go check out these gators.
I think you should.
We'll show you some gators.
I think you should check them out.
Alan, thank you as well.
Yeah, had a good time.
Alan can be found at dovedocs.com, where he blogs from time to time. How's the writing these days? Good?
Oh, going well.
Excellent.
Yeah, keeping pretty busy.
Do send us your questions, comments, what you want to hear. I think next week we should do an email show because we have 1 billion back emails and we can't seem to catch up. So we'll do that. Sound like a good idea, Alan?
Good idea.
Save that long one for when I get back.
I will save the long one, and there was also another one. You tell me if you want me to save the other one as well.
No.
Just the long one?
From Nick.
Okay.
That one.
I'll save that for you. It's only you'll be back in a few weeks. So
right.
You can send us your comments to TWiV at TWiV.tv. You could send us an audio file, or you can send us a comment on Skype, TWiV Podcast. And in fact, I was going to play one today. I didn't. We didn't have time. We got our first audio comment on Skype from Scotland. So we'll play it next time.
Can we understand it?
Absolutely. Absolutely. It's a very good question. It's short, and we will answer it next time. So, everyone, you've been listening to This Week in Virology, the show all about viruses. Thanks for joining us. We'll be back next week. Another TWiV is viral.